Welcome to the Fungisphere webinar series, brought to you by the Sydney Infectious Disease Institute here at the University of Sydney. I'm Associate Professor Justin Beardsley, and it's my pleasure to introduce these talks, our passionate team, and share some of our mission with you. We're dedicated to increasing our understanding of all things fungal, be that human health, animal health, agriculture, ecology, or even the productive and industrial use of fungi. We are seeking to uncover the threats and the promises offered by these remarkable organisms. This webinar series is more than just a series of talks. It's a platform to bring experts in the field together to share their knowledge and hopefully to build new connections. It's a testament to the synergies that exist between researchers here, but also colleagues globally. Through these talks, we share some of the incredible work being done here in the University of Sydney, but also provide a platform for international experts to share their groundbreaking research too. So, whether you're a seasoned researcher, a student, or simply somebody with an interest in the fascinating world of fungi, you'll find something of interest in this webinar series. Together, we're going to explore the threats and the promises of the fungal world. In today's talk, the ever-dynamic Aidan Kane from University of Sydney discusses his research on the antifungal potential of bisphosphonates. Bisphosphonates are widely used and well tolerated, and their re-utilisation as an anti-infective is an exciting area of inquiry. So let's hear more about it from Aidan.
Today, for our Fungisphere talk, we have Aidan Kane. Aidan has been doing his PhD with me in the lab for the past number of years. Aidan has been doing a part-time PhD, so it's been quite a few years, extended with COVID, etc. as well. But Aidan has been doing a really good job on his project, which is in our area of interest in drug synergy. And in particular, Aidan's been exploring bisphosphonates as a way of helping out azole drugs to kill pathogens. So Aidan has many interests as well as just looking at fungi, and also has done a lot of work in the area of teaching and also performance. So I'm sure Aidan will give us a really good, entertaining and educational talk. So thanks, Aidan. You can take it away.
Awesome. Thank you very much, Dee. Yes, and thank you everyone for your attention today as well. I love attention. So yes, just going to take you through a slice of my PhD. I'm probably not going to be able to go through everything, just in the interest of time. But yeah, I hope you all enjoy it. So, I mean, obviously being a tight little crowd of mycologists, I probably don't need to kind of tread through all of these stats quite as much as I usually would. But I do want to kind of point out invasive fungal infections are obviously a massive burden on human health. These are stats from Gaffey. They're saying about 150 dead every hour, more than 1.6 million people dead per year, and 13.5 million people currently affected by a really serious invasive fungal infection. And these are all extremely conservative estimates. Because the burden of fungal disease is often in low FD areas, it's likely it's way more, and it's also increased really substantially in the past few years. These are our big three. Cryptococcus, which has been a primary focus in our lab for many years now, was the #1 cause of death in gay men during the AIDS epidemic, and some species are capable of infecting immunocompetent patients as well, which is quite frightening. Aspergillosis, both invasive and chronic, presents a really significant problem, and Candida is another really important pathogen, and hospital-acquired invasive candidiasis is a huge problem. So those are the kind of dangerous, sexy invasive infectors, but topical and cutaneous infections are also a really big burden on human health. About 20 to 25% of the world is thought to currently be infected with a dermatophytic fungus. And Trichophyton indotineae is one that's really exploding right now in South Asia, causing these really aggressive infections. And as of a few weeks ago, it was now in both Australia and the USA. So, and it's not been linked to any travel to India or South Asia. And toenail infections are also extremely recalcitrant rather. Mucosal candidiasis is one of the world's most common infections, a huge burden on the clinical system. It's mostly Candida albicans, but other species are definitely emerging. The biggest one that I really want to explore is Candida glabrata, which is responsible for a lot of recurring infections. So all of this to say that there is a real need to expand our antifungal toolbox to kind of meet these growing needs. These are our current, this is from a review I wrote last year. These are our currently in use antifungals. We've got our polyenes like amphotericin B, echinocandins like caspofungin, and so on. What you might notice is that there are very few classes of antifungal, and the ones we do have are often quite toxic to the host. But the main one I kind of want to talk about today is the azoles. So this is a group of drugs that includes fluconazole and itraconazole, voriconazole, so on, used for systemic infections. But some others like ketoconazole and clotrimazole are also used for mucocutaneous infections. Azoles work mostly by targeting the ergosterol biosynthesis pathway. And ergosterol is a really important part of the membrane lipid environment. I'll talk a bit more about that in a second. Azoles work by inhibiting ERG11, which is linosterol 14-alpha demethylase, causes a buildup of toxic ergosterol precursors that are then incorporated into the membrane, causing membrane permeabilization, oxidative stress, and it's not good for the cell. The cell doesn't like it. A big part of why this need for new drugs is so urgent is this rapid spread of antifungal resistance. I'm sure most of the people here know probably more about this than I do. And I know there's a lot here, but I wanted to really highlight that Candida glabrata has a huge azole resistance problem because it's great on switching on active efflux pumps. And similarly, Candida krusei we consider it intrinsically resistant. It's not really Candida probably anymore. But due to some kind of substrate binding modifications in ERG11. Azole resistance is a really big problem in our other big pathogens as well. It usually works slightly differently. It's really diverse. In Aspergillus, it seems to be mostly driven by duplications of the target gene that allow it to create these kind of gain of function mutations quite quickly. Cryptococcus is interesting. It's not really a universal answer. Some strains exhibit heteroresistance, which appears to be at least partially driven by aneuploidy, which is really interesting because it's a haploid organism. And dermatophyte resistance is also a really big problem. Toenail infections, this sounds dramatic, but toenail infections are often considered terminal, mostly just because people end their life with them, basically, mostly due to secretion of extracellular matrix, preventing drugs from actually penetrating. So critically, I want to point out both Cryptococcus and Aspergillus are known to acquire cross resistance. That's a huge problem. And when we're kind of looking at the One Health story, they're both out in the environment. They're both being exposed to azole-based fungicides. They're adapting to those fungicides. And when it gets into a human, it's just so much more difficult to treat. And this really highlights the need to use druggable targets that actually aren't used in agriculture. So we don't just need new drugs. We don't need to keep developing drugs in the same class, but we need new classes of drug as well. So this is a small slice of the Carter crew. We work on a really diverse array of projects, virulence studies, systems biology, and we work on a ton of different fungal pathogens, which has been great over the course of my research. But one of our enduring interests is this. We look at drug-drug interactions. We love using drug synergy to improve the activity of pre-existing antifungals. And my project kind of exists down here in these two arms. Synergy can be a really great tool when it comes to repurposing and repositioning drugs. We take FDA-approved non-toxic drugs and we kind of rehome them into this new context. And this is great because it massively speeds up the really protracted drug discovery process. It's extremely expensive. I think it takes on average, I think 12 to 15 years from farm to table. And when I started, we also really wanted to kind of find ways to improve fluconazole specifically. Fluconazole's a great drug. It has great neural pharmacokinetics, which is important for cryptococcosis. It's non-toxic. However, resistance is a huge problem, and it's fungostatic instead of fungicidal, which is sometimes not ideal. So a previous PhD student in our lab, Hin Chong, did some proteomics to evaluate kind of how Cryptococcus was responding to fluconazole. The overall goal here was to kind of identify proteins that were being upregulated that were also potential druggable targets. I'm hoping that it might lead us into some rational drug design of synergists to kind of improve the efficacy of fluconazole. Diving into these proteomics, we found a few druggable enzymes that were upregulated. Well, sorry, Hin found I wasn't here. Found a few druggable enzymes upregulated in response to fluconazole compared to untreated cells at the same time point. But one of the most promising targets was this one here, which he found was orthologous to farnesyl pyrophosphate synthetase, or PPS. Now, FPPS is part of the mevalonate pathway, which is responsible for the synthesis of squalene. And squalene feeds into a bunch of different anabolic processes in the cell. He did some chemoinformatics and found that this enzyme could be inhibited by a class of drugs called bisphosphonates. So, bisphosphonates are used really widely clinically, mostly for low bone density disorders. They bind to hydroxyapatite, which is the mineral that makes up bone, where they're taken up by these cells called osteoclasts. Now, osteoclasts are cells that kind of patrol the surface of bone and break it down so that it can be repaired and remodeled. And these cells take up bisphosphonates. Bisphosphonates then inhibit FPPS, and resorption of bone is reduced. And bisphosphonates are really attractive as novel antimicrobials because they have low host toxicity, and they're also immunomodulatory. The buildup of the precursor IPP causes proliferation of gamma delta T cells and activation, which is really nice and has been implicated in dealing with fungal disease. Bisphosphonates have been explored a little bit previously as antiparasitics in protist pathogens like trypanosomes and malaria. And FPPS is, like I said, part of the mevalonate pathway. So it's responsible for the synthesis of squalene. What's interesting is that squalene is a precursor to the ergosterol biosynthesis pathway, which is the target of azoles. So knowing that FPPS is part of the mevalonate pathway, knowing ERG11 is part of the ergosterol biosynthesis pathway, if we use bisphosphonates to inhibit FPPS and azoles to inhibit ERG11, the overall hypothesis of my work is that we might be able to produce some antifungal synergy and exploit all the lovely kind of things that come with it, like reducing the required dosage of each drug, overcoming resistance, and preventing further resistance from developing. So these are the kind of three main aims of my work. I was looking at repositioning bisphosphonates that were already on the market, investigating the spectrum of activity and whether or not they synergize with azoles, interrogating the antifungal mechanism of action, and using bisphosphonates as lead compounds to kind of try and develop a suite of even better antifungals. So first off, I did a lot of MICs. A really interminable amount of susceptibility testing using, well, for those who don't know, it's a super standard technique. It just involves serially diluting the drug in media, adding a standardized amount of yeasts or spores, then incubating. What we're looking for here is the minimum inhibitory concentration, the MIC, which is the lowest point at which growth is inhibited. These are the drugs I tested. There were some others as well, but won't go into that. Generally found that risendronate and alendronate were okay, but the best one, at least in an antifungal context, is definitely zoledronate. Although it's worth pointing out, these MICs are definitely a little high, and it's kind of questionable whether or not these would be really clinically useful, but there are some specific species that really stand out. So yeah, these are all the kind of organisms I tested. So quite a broad array of clinical isolates, and a few groups stood out as being extra susceptible. VG2 Cryptococcus or Cryptococcus juderigatii, and that includes really fluconazole resistant isolates. And Candida glabrata was super susceptible to bisphosphonates, and that includes really, really massively azole-resistant isolates, things that don't have a fluconazole MIC at any concentration that we tested. And there seemed to be no correlation at all between azole MIC and bisphosphonate MIC. And that's really promising, and it kind of maybe starts to imply that zoledronate might not be a pump substrate, and that's really attractive. And the dermatophytes were a little bit extra susceptible as well to bisphosphonates. So really, they were okay at best on their own overall, but I kind of wanted to investigate that hypothesis and see if double targeting these pathways really did produce synergy. And generally, we test synergy with this checkerboard assay. It works similarly to MIC testing, but instead of having one drug in serial dilution, we've got two. So we've got this kind of a two-dimensional matrix of different drug dilutions. We have the cells, we incubate, and then we try and kind of find the point where cells are inhibited, where the concentrations of both drugs are at their lowest. We then calculate the FICI by adding the ratios of the MICs of each drug alone, along with the MICC of each drug in combination. So that's a phrase that I'll be using a few times. through the talk. So we're looking generally for an FICI that's below or equal to 0.5, which kind of tells us that there's been at least a fourfold decrease in each drug required to inhibit. So that's what we're aiming for. That's what we call synergy. And so I screened combinations of these 3 azole antifungals in basically all of the isolates that I mentioned on the previous slide. And I found that for the most part, zoledronate was again the best. It produced tons of synergy combined with fluconazole and ketoconazole. And you can kind of see that here as well. This is Candida glabrata, which is kind of uniquely susceptible, but the overall trend is the same. The mean FICI is getting kind of deeper and deeper into that gray zone, synergy zone with these 3 drugs. So it seems like zoledronate and the azoles could be a really useful combination treatment strategy. If we kind of look at the full spectrum of activity here, there are a few kind of patterns I want to point out. I'll just note that we're looking for the green cells here, which indicate a synergistic hit, kind of the greener the better. And first off, there's a bit of synergy between zoledronate and fluconazole in the cryptococci. Sorry, yeah, I knew I had a box here somewhere. Yeah, so zoledronate and fluconazole in the cryptococci. Various azoles down here in the dermatophytes. There seems to be quite a synergy pattern here. And also, interestingly, kind of basically all of the non-albicans candidas have quite reliable synergy, and there's a lot of azole-resistant isolates in here as well. So that's all starting to be really promising stuff. So kind of a caveat to all of that is that both of these drugs alone are fungostatic. It's almost like this dogma that fluconazole is fungostatic, but I mean, the reality is a bit more nuanced than that. But basically what I wanted to do was see whether combining these drugs co-potentiated some fungicidal killing instead of fungostatic inhibition. I tested this with it's a really simple time kill curve. You just inoculate media with either drug or my DMSO no drug control. Then I pulled out an aliquot at various time points, serially dilute them, backplate them, obtain a viable cell count for each treatment. So really basic stuff. So the key takeaways here are that the drugs alone are fungostatic. There is inhibited growth. But once the drugs are combined, there starts to be, certainly in Candida glabrata, once the drugs are combined, there starts to be this killing effect that just increases the more drug that we add. Again, albicans, which is consistent with the synergy data, does seem to be less susceptible to this. So the combination alone isn't enough to actually start killing the cells. But once we kind of go above that MICC, that minimum synergistic dosage, then we do start to see a little bit more killing, though certainly not to the same extent. So it's worth pointing out that even for Candida glabrata at least, even at 4 times the MICC, the concentration of both fluconazole and zoledronate are still below the MIC because synergy was so good. So what that means is that we're using a lot less drug for an increased killing effect, which is great. I'm sure I don't have to tell most of you guys that biofilms are important. They play a really important role in iatrogenic routes of infection with the yeasts, but they're also really important for filamentous fungi. Aspergillomas are technically biofilms. They form in pulmonary aspergillosis. And I mentioned before, the dermatophytes produce quite a lot of extracellular matrix in toenail infections. So that's biofilms play a big role in those infections as well. It's kind of questionably important in cryptococcosis. I'm curious to hear what people's opinions are. There's debate there. It's not as well understood. But anyway, so we want to see if these drugs work in biofilms as well. So we take the planktonic cells, we let them form these kind of stable biofilms in 96-well plates, and then we treat them with drugs. After treatment, we just use a sugar reduction assay to colorimetrically kind of measure what the metabolic activity is. And the combination definitely has improved antibiofilm activity compared to either of the 2 drugs alone. And this is true. This pattern kind of holds out in all sorts of organisms, the cryptococcuses, most species of Candida, the dermatophytes, and some of the other clinically important molds. So there does seem to be some antibiofilm activity in this synergy. Now, one of the key advantages of combination antimicrobial therapy is this kind of assumption that because we're increasing the number of drug targets, we're really reducing the likelihood of resistance developing. this, I mean, stands to reason, but very few people seem to actually really test this. So I kind of set up this really simple assay to see if this is true of fluconazole and zoledronate. I took a bunch of different organisms, cultured them in sub-inhibitory concentrations of each drug, individually or together, and I then passaged these cells in increasing concentrations of drug to kind of see if I could drive adaptation to the drug-rich environment. And what I found was that cells adapt in both albicans and glabrata. Cells adapt really strongly to fluconazole and zoledronate. It does seem that there's a loss of fitness in zoledronate at high concentrations in the albicans. I mean, we're looking at a huge amount of drug there. So we're probably looking at an osmotic effect. And the cells just can't seem to adapt and resist both drugs together. And this is true of a bunch of different organisms, basically everything I've tested. Here's a similar effect in dermatophytes, where I passage agar plugs of mold into agar with increasing amounts of drug. And you measure the radius of the colony down here. We found pretty much the same thing, that the combinations prevent resistance developing. I'll note I'm using ketoconazole here instead of fluconazole, just because it's a bit more relevant to cutaneous treatment scenarios. So, I mean, and then the big question in kind of drug discovery is whether these in vitro results translate at all into in vivo efficacy. I've used a really simple invertebrate model of infection. We take Gileria melanella larvae, we inject them with the inoculum into these nubby little prolegs, give them some time to breathe, and then give them a drug infusion. And then we monitor survival every day. This was all done with the help of the Kane lab, not CAIN, a Kane lab at Macquarie University. So what we found, I found, is that combining drugs really significantly increases its in vivo activity. Fluconazole partially rescues the larvae infected by both albicans and glabrata. But there's many more combination treated larvae still kicking at the 8-day mark, both in albicans and glabrata. And it's definitely worth pointing out that the concentrations in the combination are 4-fold lower than either drug on their own. So much less drug is much more effective, I guess. And that's good, because actually zoledronate turned out to be slightly toxic to Gileria, more toxic to Gileria than it is to humans. But I mean, that's a digression that I don't need to talk about. Shouldn't have brought it up. So that's aim one. Just to recap, we've got bisphosphonates that they are slightly antifungal on their own. They do synergize with azoles, particularly glabrata and dermatophytes. These combinations are killing. They're not just fungus static. They do have some antibiofilm activity, and those combinations also seem to abrogate the development of resistance. And it also works in vivo in a obviously pretty perfunctory invertebrate model. So now that I know that those drugs do work, I kind of wanted to figure out how. Interrogating mechanisms is great. It's kind of what got me interested in microbiology in the first place. And I'm going to be mainly discussing Candida here, but I've shown a lot of this is true in crypto and dermatophytes as well. So I'll talk more about that in a sec. So first off, I kind of had to make sure that this original hypothesis was correct, and that this synergy was due to squalene synthesis inhibition. There are a few other kind of potential cytopathic pathways that FPPS inhibition has these kind of like knock-on effects in. I performed a rescue assay where I added in exogenous squalene to see if it rescued growth that has been inhibited by zoledronate, both alone and in combination. And it sure seems to in this kind of dose-dependent manner. So cells inhibited by both zoledronate and the fluconazole zoledronate combination are able to be restored when we add in extra squalene. So that's great. It kind of confirms what we suspected. Zoledronate targets FPPS, depriving the cell of squalene. So squalene then can't feed into ergosterol biosynthesis. So, I mean, then the next logical thing is to confirm whether that was true, whether bisphosphonates affected the lipid composition of the membrane. Ergosterols, it's a really important membrane lipid. It constitutes a huge amount of the wet weight of the membrane, and it's responsible for regulating the higher order structure of the membrane, how rigid it is. And interestingly, ergosterol, along with sphingolipids, make up lipid rafts, which are these kind of fatty membrane domains that serve as anchor points for transmembrane proteins. I'll talk more about that in a sec. So I treated my yeasts with each drug, extracted ergosterol using N-heptane, and kind of chucked it into the spectro. The main takeaway here from this result is that zoledronate significantly decreases the amount of membrane ergosterol compared to the no drug control. And so does fluconazole and the fluconazole zoledronate combination. So it does seem to be zoledronate does seem to be affecting ergosterol content, although less so in albicans than in glabrata. However, in zoledronate treated cells, we can significantly restore this ergosterol content by adding in supplemental squalene. Whereas in fluconazole here, the squalene has actually very little effect. So it seems like this inhibition of squalene synthesis does have this kind of knock-on effect on the membrane sterol population. Now, I mentioned that ergosterol is important for regulating membrane structure. I kind of wanted to see what the effect of depriving the cells of ergosterol was on the structure or the fluidity of the membrane. I did this by measuring the anisotropy of this fluorophore here. So anisotropy refers to whether a population of molecules are directionally dependent. Are they all kind of facing the same way? For this assay, I'm using TMA-DPH, which kind of embeds itself alongside the phospholipids in the membrane. And we can kind of use the polarization of this molecule to kind of investigate how wobbly it is and use that as an indicator of membrane structure. And across the board, it seems that the drug treatment significantly increases the fluidity of the membrane. So ergosterol isn't being synthesized. It's not being incorporated into the membrane, and the cell loses a lot of its structure. Now, this fluidity has some kind of interesting implications, particularly considering how effective these drugs are in Candida glabrata specifically. I mentioned already a couple of times that Candida glabrata is really good at upregulating active efflux pumps. It just likes to get rid of anything that's toxic that's also a pump substrate. But pumps and other membrane transporters, like nutrient transporters, need three things to work properly. They need energy, a regulated membrane structure, and they need lipid rafts to actually localize to the membrane. So there's kind of two things that stand out there. I know already that these drugs reduce membrane structure, and if cells aren't producing ergosterol, the lipid rafts are going to be affected. And this kind of led me to wonder whether these drugs are affecting transporter activity. So I loaded up the cells with rhodamine 6G, which is a fluorescent pump substrate. I treated them and measured the efflux of rhodamine into the supernatant. And what I found was pretty cool. It seems that fluconazole is able to really cause Candida glabrata to switch on these pumps and just get the drug out of the cell. And zoledronate does this as well to a much less dramatic extent. But once we combine them, we can see that efflux is actually really enormously impaired. This is slightly true in albicans as well, but albicans is a lot less ABC transporter dependent. The main kind of takeaway here is that this membrane fluidity, due to depletion of ergosterol, seems to significantly impact pump activity, which kind of obviously causes even more accumulation of drug inside the cell, kind of increasing its antifungal efficacy. So this might be a big part of the reason why synergy is so strong in Candida glabrata specifically. I might, how am I going for time? I might squeeze through this part a little bit. I just want to point out that zoledronate also causes permeabilization of the membrane. We can look at membrane depolarization, see if solute concentration is kind of equalizing across the membrane with disc 3, which is a fluorophore that kind of accumulates and redshifts in depolarized membranes. We can also look at membrane leakage and see if basically see that nucleic acids are kind of being leaked out passively into the supernatant in drug-treated cells. But the kind of curious thing here is that while it seems depolarization seems to be kind of uniform between species, glabrata might be a little bit more affected. This leakage seems to happen way more in combination-treated albicans and also Candida cruzae, which I haven't talked about as much, than it does in Candida glabrata, which is a little contradictory considering how much more resistant to it albicans seems to be. Personally, I think it's evidence of two mechanisms, a kind of low concentration one and a high concentration one that's a little bit more disastrous for the membrane. So considering that, I really wasn't satisfied about why Candida glabrata was so much more susceptible to these drugs than the other candidas were. I decided to kind of have a look at exploring oxidative stress and see if that was part of the answer. I looked at ROS accumulation using a ROS indicator. The positive control here is hydrogen peroxide, which obviously causes oxidative stress. And I found that the combination causes significantly more ROS buildup than either drug alone in Candida glabrata only at the MIC. Which doesn't really seem to happen in Candida albicans. It also causes increased lipid peroxidation in Candida glabrata, but I kind of won't touch that in the interest of time. It's also not on the slide. But curiously, growth was partially rescued in combination treated cells in Candida glabrata only when I supplemented the media with tocopherol, which is quite a potent antioxidant. So it kind of seems like oxidative stress seems to play more of a role in Candida glabrata killing than it does in the other candidas. And I've kind of explored a few lines of thinking about why this might be. It has nothing to do with catalase expression. I've done those experiments, and Candida glabrata actually produces more catalase than other candidas do. I thought it might have had something to do with iron, because Candida glabrata has quite limited siderophore transport compared to other yeasts. They only have one siderophore transporter in their genome. But I did some experiments. This doesn't really seem to be the thing of it either. And what I did find kind of in trawling through the literature is that Candida glabrata is incapable of glutathione biosynthesis. Glutathione is an antioxidant, it's an amino acid precursor. It needs to pick up, glabrata specifically needs to pick it up from the environment, and it is in RPMI. So maybe limited membrane transporter activity that we kind of talked about with the efflux pumps might mean that also the cells aren't able to take up glutathione and maybe can't deal with it. I'm not sure how to go about testing this. I'd love to hear some theories. I assume I'll need a Candida glabrata with a recapitulated glutathione biosynthesis pathway. But what's curious actually is that squalene itself is an antioxidant, and maybe deprivation of squalene is playing a role in inducing peroxide as well, oxidative stress as well. The same also seems to be true in dermatophytes, but I might squeeze past this because I'm worried about time, because I wasn't looking at what time it was when I started talking. So just to recap on AIM 2, the bisphosphonates do inhibit FPPS, causing the inhibition of squalene synthesis. This results in the depletion of cell sterols, though they're not being incorporated into the membrane, and that has a knock-on effect in the membrane structure and causes hyperfluidity of the membrane. This hyperfluidity seems to inhibit membrane-bound transporters, especially in Candida glabrata, and this effect also seems to cause membrane depolarization and leakage. So there are holes in the membrane, but slightly more in albicans and cruzae than it does in glabrata. However, in glabrata, it does seem to cause slightly more oxidative stress than it does in albicans, which is curious. So that's kind of it for the on-market bisphosphonates. I'm really excited about the potential for these as combination therapy, but I really think they probably are only suitable for superficial infections, especially in kind of recurrent candidiasis, for non-albicans candidas, and probably for the dermatophytes as well. We actually secured a provisional patent for this combination treatment a few years ago, but that's definitely that's dead now. But actually, they're probably not going to be super useful for systemic infections. The reason for that is, unfortunately, FDA approved bisphosphonates bind to bone. We want them to bind to bone, but that really limits their bioavailability and kind of prevents them from spreading to peripheral tissues where the burden of disease is usually the highest in invasive mycosis. So this bone binding also kind of has some nasty side effects: osteomyelitis, osteonecrosis, and it has some contraindications for anesthetics. So they get in the way of surgeries and stuff as well, which is not good, because if you have a systemic fungal disease, you're probably quite sick. A few people over the years have kind of managed to get past all of this by making these lipophilic bisphosphonate derivatives. This means it's just the drug here is zoledronate, but it has this aliphatic alkane tail kind of tacked onto the end of it. And they were looking at, I mean, most of these people are specifically looking at improving its antimalarial activity. But what they've all found is that alkylating these bisphosphonates results in reduced binding to the bone mineral, increased penetration into the parasite or incorporation into the membrane, increased enzyme inhibition, quite limited toxicity to the host. And it also kind of preserves their immunomodulatory effects, which is really attractive when making an antimicrobial. So I spent 18 months in a synthetic chemistry lab trying to make this compound. I call it, we call it Tenzol because it's zelegenate with a 10 carbon tail. I learned a bunch of really cool techniques. I did actually manage to make this compound, but isolating it and crystallizing it was a process that actually ended up taking several years and a very experienced pair of hands as well. And we were never able to kind of synthesize some of the other compounds we had in mind. But in the meantime, a colleague of mine, Felsia, designed these other FPPS inhibitors chemoinformatically and synthesized them. So they're all prolyl aspartic acids. So it's kind of like a dipeptide. We've got the aspartate residue here, the proline residue here, and it has these lipid tails. So yes, and those were all kind of predicted to bind to the FPPS active channel, or substrate binding channel, rather. So derivatives were tested for activity against 8 clinically relevant fungi. And we got the MICs. I also kind of investigated whether they were toxic to human HEC cells by obtaining just the IC50 with a simple cytotoxicity assay. And what I did here is I kind of took the ratio between the geometric mean of the MICs in fungi and the IC50 in human cells. Positive ratios then mean that they're kind of good candidates because they're more toxic to the fungi than they are to humans. And these ones are the bad candidates because they hurt us more than they hurt the fungi. What you might notice is that Tenzol is kind of the only good derivative that we tested. It's really exceptionally promising in that respect. It's much more toxic to the fungi than it is to humans, more so even than zelegenate is, and that's extremely non-toxic to the mammalian cells. So that's really exciting stuff. Interestingly, these prolyl aspartic acids, there's some synergy. None of it is really sufficient to bring the MIC of these prolyl aspartic acids down below the IC50. There's an exception, but it's only slight PDA12. However, Tenzol, there is some interaction, a little bit of synergy, not quite as much, certainly not as much as zelegenate. But I mean, the dosage of the drug that's required to actually inhibit the cells is just so much lower. So it's still a really attractive lead. So these are the Tenzol MICs in blue here compared to the zelegenates in yellow. And it turns out that they're on average at least 100-fold lower than zelegenate, the MIC. So there's a 100 to 200-fold increase in activity just by adding this 10 carbon tail. It's got an extremely broad spectrum of activity. I'm sure you can see that here. It is quite active against a huge majority of the clinically relevant fungi that I tested. And definitely less synergy than zelegenate overall, but I mean, it's such a good drug, it doesn't really actually seem to matter. I'll also point out that Tenzol itself is fungicidal, not particularly fungicidal in Candida albicans at the MIC, but as we kind of increase it above that, we do get this killing effect. Whereas in Cryptococcus, it appears to be fungicidal at the MIC, which is really nice. And this is a bit of a slapdash side, I apologize for this. But basically the mechanism appears to mostly be the same. It's governed by Tenzol inhibits squalane synthesis. It causes depletion of ergosterol, hyperfluidity of the membrane. That causes kind of depolarization. It causes leakage and oxidative stress in all of the species that I tested. The last thing that I kind of wanted to show is that Tenzol does work in vivo as well. I used a really nasty strain of Candida auris for this. It's a really aggressively virulent strain. It's basically pan-resistant to all of our kind of known antifungals. What we can see is that Tenzol significantly rescues larvae after just one infusion. A lot of people do multiple infusions to kind of buff up their data a little bit. But there's a really significant increase in rescue after just one injection. And zelegenate is actually fairly, like I said, fairly toxic to Galeria at around the MIC, which is not good. But there was little to no toxicity of Tenzol in Galeria at any of the concentrations tested. So that's really attractive as well. Really promising. So just to recap on that final aim, we have those prolyl aspartic derivatives with those little alkane tails. They're not good. They're more toxic to to human cells than they were to any of the fungi. However, this lipophilic derivative of zoledronate is definitely much more attractive. It's way better than the native zoledronate. Quite low human toxicity, a little bit, maybe more than is completely desirable, but it's quite low. It seems to basically retain the same mechanism, and there's and it definitely works in vivo as well. So that's really great. So just to kind of recap everything, our on-market bisphosphonates might make for really attractive adjunctive therapies, probably mostly for topical infections though. The antifungal mechanism of these drugs seems to be mostly due to squalene synthesis inhibition, which kind of has these knock-on membrane-related effects. And finally, I've shown that an alkylated version of zoledronate, it has massively improved in vitro antifungal activity and in vivo. So yeah, thank you to all these wonderful women who have helped me over the course of my research. The Kane lab, Hui and Amy in the Kane lab at Macquarie helped me out with my Galeria infection models. Jane and Felsie were both instrumental in synthesizing our novel compounds and helping me to synthesize them, and then when I failed, basically coming in swinging and succeeding. And of course, all of these people in the Carter lab have had a hand on this ball over the years, and my wonderful, lovely patient supervisor, Dee Carter. Thank you to you as well. Yeah, and thank you so much for your attention. And then I'm going to just go through all my extra dummy slides that I have in case of emergencies.
Thanks, Aidan, for a terrific talk. Do we have any questions for Aidan?
Yeah, I do. Firstly, I just want to say that just blew me away. I had no idea you were so far advanced with this work, and it's beautiful science, and it's just really nice, and that to see you come this far, and I hope you're really proud of the stuff you've done. It's really, really good. So sure. I got lots of questions, but maybe one day I'll have to just come over and talk to you at lunchtime. But sure. I just wanted to ask, with the Galeria, when you showed the initial synergistic effects, I can't remember which fungal model you used. And how many fungal models do you have set up with Amy? Because currently we're just looking at Cryptococcus, and sounds like you've got a Candida auris model going as well.
Yes. So sorry, I'm looking at the wrong thing now. Yeah, go. Yes.
Oh,
so
yeah, there we go. So you're using glabrata and Candida albicans. I didn't know that there were glabrata models around. So that's fantastic.
Yeah, it needs a really high multiplicity of infection. You need to put tons of yeast into the larvae to get them sick. So I think it's like maybe 50-fold higher in glabrata than albicans.
Okay.
So yeah, it's definitely not great. I mean, in an ideal world where we have infinite access to larvae, we would probably, I would really like to do a mixed model of infection. Whether or not Candida glabrata on its own is able to establish real disease anyway is a bit questionable. It seems to like surf Candida albicans like down to penetrate the tissues. So I think a mixed model would be better to really kind of understand the in vivo applications of this for treatment.
But you've got they've got Candida albicans and Candida auris going, which is really great. So
yes, so we set this
kind
up.
of paved the way for anyone else coming along, because I'm sure that they've already worked out the MOI and everything. So
yes, I did that. So we did a tolerance test basically to see what was an acceptable amount of what the acceptable MOI would be, and auris is low for this particular strain. It's a really aggressive aggregative strain.
Yeah. And have you got any plans to go into mice to test these synergistic effects, or are you just more now sort of resigned to the fact that because of that toxicity that you really only want to test it in superficial infections?
Yeah, yes, the latter. I mean, there are all sorts of great tools to kind of look at in vivo mucocutaneous treatments. I actually applied for a grant a few years ago that I unfortunately didn't get, to buy L'Oreal actually makes these little pellets of tissues. They're these kind of reconstituted epithelia, and they have various. They have oral epithelia, vulvovaginal epithelia, and skin like epidermis. And I kind of wanted to try and infect those. Those have kind of been used as a bit of a step up in vitro model to see whether or not infection works, but they're quite pricey and they're very small. So unfortunately, we couldn't quite get those. I think that's what I would want to do with this, with this work. Well, I say that I personally am ready for the next thing after all these years, but yeah, I think that would probably be the next step for this arm of the project. Yeah,
it's really nice, Dee, how you've gone from you know, the basic science discovery back in 2012, you know, seeing that go up with the proteomics and yeah, and then taking it to this level, it must feel really rewarding to do that.
Yeah, it's quite amazing actually, because so many of these things that look promising in an omics test just fall over, you know, so it was really nice to see that it actually sustained and was useful.
Yeah. You actually got some real data, you know, some data that meant something.
That's right. That's right. Thanks to a lot of hard work on behalf of Aidan and a few honest students on the way as well.
Yeah, it's a nice proof of. I mean, I just kind of have been writing about this in my discussions at the moment. It's a really nice proof of concept. I mean, I think so often rational drug design it doesn't really bear the fruits that just those massive screening studies do, where you're looking at 120,000 compounds or whatever, and seeing whether or not you ping any hits. And so it's nice. Yeah, I'm definitely grateful that this.
And sorry, I know I'm hogging questions, but I think I asked you this before. It's really interesting that you get a synergistic effect, because you think that you're targeting two points in the same pathway. And so, you know, initially you would think that you wouldn't get a synergistic effect, but I think I asked you this once at a talk you gave. I think it was a yeast meeting in Brisbane, and I think you pointed out that the squalene synthesis is sort of earlier on in the pathway, and that so there's a bifurcation that occurs before you get to ergosterol. So in fact, you're sort of inhibiting the same pathway, but it branches. Is that correct? So you're sort of blocking two pathways.
Yeah, definitely. So yeah, squalene has quite a few uses. I'm also convinced that the antioxidant properties of squalene are definitely part of it. And well, I kind of mentioned this briefly. FPPS does also kind of have its own kind of branching pathways as well. I think I'm pretty confident that I've shown that those branching pathways aren't really critical to the antifungal effect, but I'm sure something's happening. Those that I kind of mentioned, protein prenylation and stuff, those are really important for the antiparasitic mechanisms and the anti-cancer mechanisms. But that's not kind of what I found in the fungi. So maybe there's something happening there as well.
So there may be an off-target effect as well, potentially.
Yeah. It's definitely possible.
Which there always is. I mean, you know, everybody likes to think you've got this drug against this, you know, this one pathway, but really all the drugs that are approved out there, I'm sure, well, you know, we've seen it with statins as well. I mean, I think there's an alternative target other than inhibiting cholesterol synthesis. So I think if we knew everything about our drug, we'd never ever get them FDA approved. So,
yes, definitely.
Yeah, no. Well done.
Thanks.
Really good.
Thanks, Julie, for
Thank you, Julie.
questions. Does anyone else have questions they'd like to ask?
Hi, Aidan. Thanks for the interesting talk. You already mentioned a bit when you were addressing Julie's questions. So you are just focusing on topical administration because of the toxicity. Is that right?
Yes. So that's for the combination treatments, the FDA approved ones. I think they're probably only going to be useful for topical treatments. However, I didn't go super deep into this the way I probably should have, but the lipophilic derivatives have been shown to have reduced bone binding. So hopefully that has a bit more legs as a systemic treatment.
Right. Yeah, because normally when people give zoledronic acid for osteoporosis, they do it by intravenous injection. because the drug has very low oral bioavailability. So I'm wondering whether your analog will have a better PK profile like oral.
Yeah, it's a good question. I'm not sure. So I know alendronate has better oral PK, and there was some synergy observed as there as well. But yes, so the very similar compounds have been explored in a looking at a like late stage liver growth rather in malaria in a murine model, and they did find they gave it by no, that was intravenous as well. Actually, I was there was something about oral the bioavailability of those that was that's just stuck out in my mind. But it's certainly not confirmed.
Okay.
Either way, I think that would be the next step. Even just to see whether or not it is actually binding to the bone, having an idea of the PK is definitely the next step, I think.
Yeah. Okay. Thank you.
All good.
Yeah, I might just also say we've talked to a colleague who's got a lot of interest in bisphosphonates, and he feels that maybe there's the potential that they bind to the bone, but then they leak out slowly, and that could actually be a good point of their binding, is that then they sort of maintain this lower but sort of consistent level of sort of leakage back into the peripheral tissues and stuff. So that's something that we would love to explore if we had more money and time.
Like a reservoir, right? You have a reservoir.
Yeah, like a reservoir, exactly.
It's like an implanon, kind of.
Yeah. And because they're not very toxic, you could potentially load up to a certain extent and hope that you would get enough coming back out that the synergy would work. But yeah, something to think about for the future.
Yeah, probably someone else's problem. I mean, the unfortunate thing is that I mean, we talked a few years ago with some colleagues in pharmacy about making these radio salts using the cyclotron under RPA to kind of load them up with technetium and actually track where it is in the rat in a I think it was a rat model. But you just need like tens of grams of drug to be able to even start that stuff. And our synthesis process was really fraught, and our colleagues have kind of big doubts about the scalability of it. I mean, it took 4 or 5 years to get a gram. I think less than a gram. So that's something that's a bit of a caveat to all of that, is that the synthesis process is really imperfect, and it'll take quite a bit of work to get it good.
Can I just a very quick comment? I don't have any intelligent questions to ask. I thought it was a great presentation, though. It's really like beautifully presented, very clear, tells a great story, and it's just interesting to hear about the process, even of like repurposing drugs and how complicated it is, and getting them over the line and all the work that goes into it. So thanks very much for sharing it so clearly. I really enjoyed it.
Oh, of course, yeah. Thank you for having me, Justin. Yeah. Thank you.
All right. If there's no further questions, thank you again, Aidan, for giving us that very lovely talk.
Thank you,
Thanks
everyone.
everybody for turning up to listen to it.
Thank you for joining us. I hope you've enjoyed today's talk from the Fungisphere webinar series, and we look forward to seeing you next time.